Hello and welcome back to Microbiology. We're going to cover today Chapter 15, which is going to be a continuation from Chapter 14. Again, in Chapter 14 and 15, we're covering immunology. So in Chapter 14, we covered the first and second line of defense. All Chapter 15 is going to be about is the third line. Specific immunity has another name, and that is acquired immunity. And the idea here is that we acquire more and more immunity or more memory as we, again, have more need. But it's important to understand that since this is specific, that we need the ability to basically react to lots of different things. And that's where the word immunocompetence comes in. But what do B and T cells respond to? Well, the answer is antigens or immunogens. Antigens and immunogens are chemical substances that stimulate the response of B and T cells. These are again usually protein or sugar materials that are found inside or outside of cells or viruses. It's important to note that usually your own antigens do not trigger your own immune system to respond against them. But there are times where we do see situations where a person's immunity starts attacking self, which we often refer to as an autoimmune disease. The important characteristics for this adaptive or specific immunity is really the fact that they're specific and adaptive. With specificity, again, the specific response to a single antigen, this makes this very directed, meaning that we're going to only target and fight one thing for each of the B and T cells out there. And so the idea here, again, is we're very specific. But if I only had the one cell, that wouldn't help me a lot. So an important feature is that adaptability, because once we've found a threat, we would keep more cells around in the future to fight that same threat. And that's what memory is. The ability to basically rapidly mobilize lymphocytes that have already been exposed to the first encounters of a specific antigen. That way we're more prepared to fight that antigen again in the future. So whenever we look at this, lymphocyte development is going to have basically some core characteristics in how we made white blood cells. But really, where this gets interesting is whenever these lymphocytes are presented with an antigen, at which point B and T cells do different things. The B cell, whenever it becomes presented to an antigen, starts to produce what's called a plasma cell, and that plasma cell pumps out antibodies. So how B cells actually fight is antibodies. The T cell, on the other hand, whenever they're challenged or presented with an antigen, they reproduce and can ultimately then go producing cells that then fight other cells. So we call it cell-mediated immunity. So here's an illustration of exactly what we talked about. Again, the B and T cells are produced the same rough way that other blood cells are produced. But there is a major difference, and the difference comes then in the maturation process, because B and T cells have to mature at different locations. B cells will mature still in the bone. T cells mature, on the other hand, in the thymus. So B cells mature in the bone, T cells mature in the thymus. But once they've matured, then we let them out to go look for the thing we're looking for. And so they go to places like lymph nodes and spleens. So let's show you what happens whenever we activate or present an antigen to B and T cells. So notice here we're looking at the introduction of an antigen to a dendritic cell, and really again this antigen would be picked up whenever we destroyed the cell that had it. So what happens then is the dendritic cell can present the antigen to other cells like B and T cells. So notice in this example we have the dendritic cell, the specialized macrophage, presenting the antigen of something bad to the T cell. And in this set, this is a T helper cell, which then can go help activate other parts of immunity, including B cells. Once the B cell has been activated, notice that the B cell can really turn into a couple different types of B cells, which includes memory B cells, which will hang out for future encounters, and then plasma B cells. Those plasma cells again pump out antibodies, which is how we fight. But notice what happens with the T cell. The T cell again can turn into things like, or can lead to the activation of things like memory T cells, Or we can continue down this path, which we'll show you on the next slide. So notice here we have this scenario where the T cell again turned into a memory T cell, but it can also turn into things like a T helper cell, a regulatory T cell, and a cytotoxic T cell. Back over to the B cell side, notice how again the plasma cells pumped out antibodies. Again, the antibodies are what go fight, and we refer to the fighting with antibodies as humoral immunity. Since we're going to fight with cells over here, we call this again cell-mediated immunity. By the way, the big effector cell here is a cytotoxic T cell, which is real good at killing other cells. So let's talk a little bit more about how we can recognize things as self and non-self. We've talked about antigens at this point, but we need to go a little deeper. We need to point out that there are some very specific immune system markers that we use for recognition of self. And so we call these markers the MHC, or the major histocompatibility complexes. Basically, interactions with MHCs can allow us to understand what is self and what is not self, creating then a response. So there are really going to be three different types of major histocompatibility complexes: Class 1, Class 2, and Class 3. I really care more that you know about Class 1 and Class 2. With Class 1, we're going to see these markers are going to be present on all nucleated cells. So Class 1 MHCs are going to be found on, again, all nucleated cells. This is really, again, illustrating self. So MHC Class 1s tell us what belongs to self. Naturally, again, if we see an MHC Class 1, we should not inherently attack that MHC Class 1. Whenever we talk about Class 2, this is going to have a very specific role. If you recalled a few slides back, I showed you where a dendritic cell presented an antigen to a T cell. But notice how the T cell didn't attack the foreign antigen, or the thing holding it. And it's because the way the dendritic cell presented the antigen. It presented it on a marker called a Class 2 MHC. And so what happens is the Class 2 MHC is going to allow for the showing off of a foreign entity without eliciting response against the cell. So I can warn you about what's coming, and you're not going to kill me thinking I'm that bad thing. And so these are again presented on special types of cells we call antigen presenting cells. And it's really just three types of cells: macrophages, dendritic cells, and B cells. Naturally, again, these cell types would be involved in presenting antigens to things like T cells. MHC Class 3 I'm not so infatuated with here, because again, it's going to play a role in the complement system, and we just briefly covered the complement system. So again, you should know MHC Class 1 is indicator of self and found on all nucleated cells, and then MHC Class 2 found specifically on macrophages, dendritic cells, and B cells, and is really allowing for presentation of antigens without eliciting immune response. So notice on the left, we have a cell that has an MHC Class 1. What should that suggest to you about this cell? The answer is that it has a nucleus, and that's part of self. But what's going on over here where we have both an MHC Class 2 and an MHC Class 1? Again, the one told us it's self, and that again, it is part of us. But antigen binding site on the MHC Class 2 is used to present something that doesn't belong, or something foreign. And so this is again the idea that we can show off foreign entities and still indicate that we are part of self. So let's move on to CDs, or clusters of differentiation. A CD is going to be important in terms of helping for recognition with immunity. There are lots of CDs out there. There are well over 300 that have been recognized. But really, I want you guys to know just about a few. Before we talk about CDs, let's show you a feature on B cells. With B cells, we use a certain receptor called the B cell receptor as our way to recognize antigens. So again, antigens would bind at the antigen binding site and activate again response because we bound the receptor. But notice, in the case of the T cell, we have a receptor, but it's less effective in recognizing antigens. And as a result, it needs additional resources like CDs to help with identification. So really a CD3, I would like you to know, is kind of basically an adapter to help hold other CDs in place. But really CD4 and CD8 is what I care more about. CD4 is going to be very important in recognition of, again, antigens of self. CD8s are going to be very important in recognitions of, again, self, but in cells that are wanting to die. So what I'm getting as CD4 T cells are not the cells that are actually going to kill other cells. The CD8 T cells, like the cytotoxic T cell, are the ones that should kill cells. So we talked about the ability to present antigens by macrophages, like dendritic cells. The idea is, again, the dendritic cell would come along a pathogen and eat it through phagocytosis, at which point then it would take and grind up the pathogen and then put all the antigens in on MHC class 2s and present it to other cells. So again, there are really just three antigen presenting cells out there: dendritic cells, macrophages, and B cells. Ultimately, whenever again the antigen activates B or T cells, they're going to proliferate and differentiate. We call this then, again, challenging cloning. And so again, cloning is the proliferation of a particular lymphocyte. It is identical genetically to its other cells, and some of them will remain as simple memory cells. Whenever we talk about how T cells respond to antigens, it's important to understand, again, there are three main functional types of T cells. The helper T cells, which help activate other parts of immunity, like activating macrophages or even activating B cells and cytotoxic T cells. We also have regulatory T cells, and as the name suggests, it helps regulate T cell response. But the last type of cell here I'm concerned with is the cytotoxic T cell, which is ultimately the cell that's going to destroy cells that are infected or cells that do not belong, so foreign cells. Whenever we talk about B cells, though, again, B cells respond very differently. Whenever a B cell is activated by an antigen, the B cell will divide and give rise to plasma cells. Plasma cells would be, again, the effector cells. The plasma cells are ultimately going to allow for the production of antibodies, and those antibodies are what's going to circulate and look for the antigens for which they bind to. So what markers do we see on T cells? Again, things like CD3, CD4, and CD8. I told you before that CD3 is really going to help assist in binding other CDs. CD4 and CD8, though, are what we're more concerned with because they're co-receptors. What a CD4 does was, again, bind to an MHC class 2, because CD4s were used for recognition of a foreign antigen that was presented by an antigen presenting cell. CD8s, on the other hand, were tied to, again, cytotoxic T cells, or the T cells that are going to recognize self, but again destroy the cells that do not belong. So let's point out that T cells, since they have to actively go hunt infection, will typically circulate. And they'll circulate in and out between, again, circulation and the lymphatic system, again, moving through places like the lymph nodes and spleen. The idea is that lymph nodes become a common meet and greet area for the T cells to encounter potential pathogens. Roughly, we estimate that there are 1 billion T cells that will pass between the lymphatic system and the general circulation per day. So we have lots of these cells moving in and out and all around, trying to look for the things that they need to destroy. B cells will mature in the bone marrow, but once they mature, they will typically head to a specific site, usually in things like the lymph node, spleen, or other lymph tissues, and then they'll just hang out. Because again, they don't have to go fight. Instead, they produce products, antibodies, that they'll use to go fight infection. So here's an illustration comparing B and T cells. You can take a look at this. So let's talk for a second about how each naive lymphocyte bears an antigen receptor that recognizes a unique antigen. The idea again is that the lymphocytes are born just like other blood cells from the hematopoietic stem cell. But what I've told you so far is that our lymphocytes need to be very specific. So it's important to understand that these stem cells give rise to basically the lymphocyte stem cell. And the lymphocyte stem cell then can become either B or T cells. Ultimately, though, an important part of this is the maturation process. B cells will stay in the bone marrow, T cells will head to the thymus. And ultimately, each will build a unique antigen receptor. Ultimately, the B and T cells have to again be very specific. Some cells are ultimately going to not be able to be allowed to leave, while other cells will be allowed to leave as they've proven that they're effective. So by the time B and T cells reach their lymphoid tissue, each one is equipped to respond to a single unique antigen, and they're ready to find and bind to those antigens. So we see the exact illustration of what we just talked about. Notice again the production of the lymphocytes happens in the bone marrow just like all blood. But then again, the lymphocytes need to go mature in their maturation places. Again, B cells mature in the bone, T cells mature in the thymus. Once they've again matured and developed an effective receptor, like the T cell receptor here, or again the antibody-based receptor of the B cell, then they will effectively travel to their desired location and then set up shop. Don't forget though that T cells will often want to circulate and look for threats, while B cells will often stay put. So how do we end up getting this kind of variability in the antigen recognition side? So this is an illustration of how it works in B cells. Notice that we have a whole section of the genetic code here, but each part is responsible for a tiny piece that could make up the pieces for recognition in the variable region of the antibody, or in this case the B cell receptor attachment site. And the idea here is that by choosing different combinations of genes, we get different combinations in that variable region. That means that each, again, B cell can have a unique pathogen it's looking for, or unique antigen it's looking for. And so again, this is genetic reshuffling recombination idea that allows us to get this level of diversity. And so the idea is that hopefully if you're developing a healthy immune system, you would have a whole robust set of all these random combinations to basically find and seek out any antigen you could encounter that's not self. The B cell receptor again is really composed of two major parts: the heavy chains, which are the long ones, and the light chains, which are the short ones. But notice really the information that's important here is the variable end, where again the antigen will bind. And as a result, again, this is the specific region. But notice how these are identical to one another on the same receptor, as will be the receptors on the same B cell. So all of the B cell receptors on one B cell will have the same variable region. And so the idea is we could bind two different antigens that are the same type at the same time. Okay? So that antigen binding site, again, specific to the thing that we're looking for. And in this case, again, it's the antigen. So that variable region has the antigen binding sites, which again can be very diverse based off of the amount of genetic material that was expressed and in what combination. The other part of that, though, the non-variable region, we call the constant region, which should not change at all. The T cell receptor, in terms of how it's kind of unique, is put together the same way. Again, this constant region versus variable region. But one of the bigger things is, again, it's not as effective. It's relatively small, and it's not going to be basically secreted. And so the idea here is the T cell receptor often, again, needs that help and recognition of other features, and that's where those CD4 and CD8s came in. So notice in clonal selection, we're ultimately trying to produce all these different combinations through random rearrangement of the genetic information in terms of what's being expressed, and potentially we could produce a receptor that would recognize self. If we were to let this loose, now we have a situation where our immune system is attacking self, which we call autoimmune disease. So naturally, if again one of the random combinations does attack self, that means we need to destroy that or delete it. Again, we call this clonal deletion. So again, we're trying to develop this tolerance to self, the idea that we remove any dangerous clones before we let them loose. And that way we don't, again, attack the self by the immune system. So whenever presented with an antigen, it should elicit a response. So an antigen, again a substance that will provoke an immune response, will be perceived by the receptor. But one of the things that you probably don't know is there's different levels in terms of antigenicity. So how antigen is the antigen. And the idea here is the more clearly something doesn't belong, the more aggressively we will attack it, or the more we'll activate immune response to try to remove it. And so that's really what antigenicity is. The idea that, again, the more clearly something doesn't belong, the more effective it's going to be in activating B and T cell response. So here we can see examples of, again, the idea of how antigenic is the antigen. And so if it's something that clearly doesn't belong to you, it's considered to be a good antigen or immunogen, illustrating that's going to elicit lots of immune response. And so you can see, again, something that doesn't belong to you elicits huge response, like bacteria and plants and human cells or animal cells that are not your own. But if it's just a small piece of something that may be a product we would make, that should elicit much less response. And as a result, we're less likely to kill ourselves or products that we're purposely making. So again, things that are more clearly do not belong elicit a greater immune response. So what are things that can be considered antigens? Well, basically most products that come from a cell. Proteins, or polypeptides, lipoproteins, glycoproteins, nucleoproteins, sugars or lipopolysaccharides. Again, a lot of components that are produced by cells that are either in or on cells can be recognized as antigens. One of the things to note, we can make a more effective determination of whether something belongs to us or not based off of its size. And if this something is pretty small, it's hard to make an effective determination of whether it's self or not. And as a result, it's considered to be a weak antigen. While things that are much bigger, we can more effectively recognize as whether or not it belongs to self, and as a result, can elicit a greater response. Epitopes and haptens are something we should probably talk about. Epitopes is a portion of the antigen molecule that's recognized and responded to by a lymphocyte. The idea here is this is what we're actually looking at to look and see if it's self or not. This is the signal point for whether something is foreign. Haptens, are going to be much smaller, and so themselves typically won't elicit immune response. But if linked to a carrier group, and combined molecule develops immunogenicity. The idea again could lead then to immune response. Alloantigens are another important topic we should point out. Alloantigens are going to be protein products that are actually made by someone else. And as a result, whenever your body sees them, we recognize it as not being self. Even though, again, it is a liver cell, it's not our liver cell. So if you think about it, remember way back whenever we talked about MHCs or major histocompatibility complexes? This is a great example of how we can recognize, again, self versus not self. We've discussed in the past this idea that toxins are going to overstimulate immune response. The body freaks out whenever, again, it sees a toxin. So things like bacteria toxins often will be potent stimuli for T cells, which allows for significant amounts of T cell activity that can overwhelm the body and cause death. Toxic shock syndrome is a great example of a certain autoimmune disease associated with having toxins as antigens. So let's go through the role of antigen processing and presentation. So we've talked about, again, macrophages, B cells, and dendritic cells as being options to present antigens to other cells. So let's go through the example here of how specifically this dendritic cell will take these microbes and turn them into antigens to activate other immune cells. So notice again the microbe is recognized as being foreign typically by things like PAMPs, at which point again the microbe is brought in through endocytosis, and the corresponding vesicle it's held in is called the phagosome. But if you recall, we had to combine the phagosome with the lysosome to make the phagolysosome, which allowed for the destruction of the cell. So we get all these little pieces of the cell that we're now calling antigens. But in order to present these antigens to activate other cells, we need to present it in a very special way. If you recall, again, that presentation had to happen on an MHC class 2. So notice how we're taking the antigen and presenting it on an MHC class 2. That allows us then to activate other things like T cells to allow them to activate other aspects of immunity. And so you can see here we've activated the CD4 cell. That leads to, again, the development of T helpers. There are several different T helpers here. Th2s are going to help activate things like B cells. Another note here, notice how B cells can activate even then T helpers, because again B cells can present antigens as well. Notice Th1s typically produce tumor necrosis factor that help again stimulate macrophages. We also have Th17, which promotes things like inflammation, and then regulatory T cells actually to help bring down again the responses of the immune system. But really, we should probably point out what happens whenever we activate a CD8 cell. That CD8 cell, whenever it becomes active, again can become what's called a cytotoxic T cell. And this is a cell that does the killing. The idea is a cytotoxic T cell has again a CD8 that's going to recognize the antigen, but whenever it's presented on MHC class 1. By the way, if a cell presents an antigen on an MHC class 1, it is eliciting that it wants to die. And the cytotoxic T cell is more than happy to offer that for that cell. And it does so by producing two enzymes, granzyme and porphyrin. The idea here is, again, the granzyme and porphyrin will lead to the death of the cell as it promotes apoptosis. So you can see here a comparison between things like the different types of T helpers and the, again, different types of CDs we have. So T helpers will often have, again, CD4 receptors used for recognition of MHC class 2s. Cytotoxic T cells, though, again, are specifically CD8s used for recognition of MHC class 1s. Notice again, memory cells can be both CD4 or CD8. Again, memory cells that are activated as CD8s can become cytotoxic T cells. And I don't care much about the gamma delta T cells. So, as you can see in this scenario, we're going to go through the activation of B cells. So notice B cells can be activated by, again, the introduction to a pathogen. So again, B cells have the ability to grind up a pathogen and present the antigens to activate T cells. So yes, T cells and B cells can activate one another. But again, whether it's activation by a pathogen or by T cells, we can have an activated B cell. That activated B cell again can proliferate and differentiate into things like regulatory B cells that help regulate immune response. Memory B cells, which again stick around for future encounters, but really the effector cell is the plasma cell. And what the plasma cell does is it becomes an antibody factory. It is literally just a protein factory here to pump out antibodies. And if you recall, again, the antibodies circulate, which we call, again, humoral response, and they'll go find and bind to the antigens that we're looking for. We just got done talking a second ago about, again, humoral response by B cells. But let's talk a little more about cell-mediated response by T cells. Again, this is called cell-mediated because we directly need the involvement of T cells for this to happen. This is very complex and very, very, very much still a bit of a mystery to us. I want to point out, though, some things that we do know is that T cells will require some recognition of self. So one version of the MHCs before we'll actually do any type of activity. We also see production of cytokines that allow for lots of other effects to occur. So let's talk more about T helpers. T helpers are going to be very important in allowing for the activation of other aspects of immunity. So they play a central role in regulating immune reactions to antigens, including again B and T cell responses. They're also going to be necessary in activating things like macrophages, or even again activation of potentially things like cytokines and interferons to prime other cells to protect themselves. Ultimately, what can happen though is the stimulation of a T helper CD4 cell can ultimately allow for differentiation into T1s, T2s, or T17s. And the idea is again each one of these T helpers having a unique feature that they can do. The T regulatory cells, or reg T cells, are going to be very helpful in helping to regulate what's going on in terms of inflammatory response, as well as basically T and B cell response. The idea here is they're necessary to help control the levels of immune response. We showed you a little while ago how again inflammation or even potentially immune response can get out of control and could kill a person. And that's the beautiful thing about regulatory cells. Regulatory cells can help regulate immune response. Regulatory B cells, again, can help regulate to some degree the response from T cells. So again, B and T cells, regulatory B and T cells, can help regulate overall immune response. So let's talk more about the cytotoxic T cell. The cytotoxic T cell has the ability to kill a specific target. So again, cytotoxics or CD8 cells are going to be able to kill other cells, which is very helpful for us. There are again the killer Ts, cytotoxics, that are going to create then a hole in the actual target cell by releasing porphyrin whenever they're activated. That porphyrin then allows a secondary enzyme to come through called granzyme. And that granzyme actually then allows the cell to go through apoptosis. So we punch a hole with porphyrin, and then granzyme goes in and causes the cell to kill itself, which usually involves dumping of its lysosomes, which then again dissolves the cell. Ultimately, it's important to understand that T cells, cytotoxic T cells, have some pretty particular targets. Cytotoxic T cells are most recognized for fighting things like virally infected cells that are even our own cells. Even versions of our own cells that are cancerous in nature can be destroyed by T cells. Or even, again, cells from another animal. Whenever we talked about B cell receptors, we pointed out it had very specific antigen binding sites. But we also point out that B cells were responsible for producing antibodies. The antibody looks just like the B cell receptor for which it came from. And so the idea is whenever again a B cell becomes activated by an antigen binding to its B cell receptor, we then see the B cell turns into a plasma cell that then can pump out more B cell receptors, but now as something called antibodies or immunoglobulins. The idea is we'll have the same variable region, which means we attach to the same exact antigen. There is going to be some wiggle room for the antigen-antibody interaction, and so the idea is that we can get partial fit that results in weak activation, or if we have full fit, we have full activation or strong activation, but if there's no fit, there will be no activation. But let's talk now about how antibodies really function. To make it super simple, all antibodies actually do is stick to antigens. And again, it's the exact antigen that the B cell from which it came from was looking for. Okay, so notice again, there are outcomes though based off of how the antibody attaches to the antigens. So notice in this situation, all we did was cover the cell with a bunch of antibodies. But by covering the cell with antibodies, that now promotes things like phagocytosis by things like macrophages. We call this opsonization. But even just covering the surface of the cell, that actually lowers the likelihood of interactions between a cell or a virus, in this case, from coming into contact with cells or tissues. And the idea is that neutralizes that cell or virus. So again, neutralization is blocking up the cell or virus's surface to prevent it from interacting with other things. That's why it's called neutralization. But again, notice the antibody, just like the B cell receptor, was again a Y shape with again two identical variable regions, meaning that you can attach to two different bacteria that have the same antigen at the same time. And so what you can end up doing is sticking a bunch of bacteria together in a process called agglutination. The next one that we should talk about here is the activation of things like complement. Again, we showed you before in the previous chapter that the presence of an antibody could activate the formation of membrane attack complexes, like complement. And the last thing here is basically neutralization of a toxin. Again, if we surround a toxin, that product can't interact with cells and thus can't hurt cells. And as a result, we call this an antitoxin. One of the things I should point out is there's actually different classes of antibodies or immunoglobulins. The idea is, again, the different types of antibodies or immunoglobulins serve different functions. A good example is something called IgG. IgG is actually one of the most effective of the antibodies that we see. It's typically a monomer, meaning it's just one single Y-shaped structure, but it's very good at finding and binding to things and circulates quite well. IgA is actually going to come as monomers or dimers, but by far really what's most important about IgA is it's often going to be secreted. So this is a secreted antibody that again would survive in environments like the mucous membrane. The other thing I should point out about IgG is its long-term immunity. This is the memory antibody that would stick around for long periods of time. Let's move on now to IgM. IgM is going to be a pentamer with 5 different Y shapes that again will be able to bind up lots of different structures, whether they be cells or viruses, at the same time. And so this is actually one of the first antibodies produced and has the job of trying to agglutinate or grab up lots of bad things. IgD is another of the different types of antibodies out there. What's important to understand about IgD is it's just the receptor on the B cell. It can be produced and secreted, and we're not 100% sure why. IgE, on the other hand, has a very specific function. This is the antibody associated with antigens and really eukaryotic infections. Did I say antigens? I meant allergies. So allergies and worm infections. So eukaryotic things. I just told you a second ago that IgG is long-term and will stick around, remembering the past infections. And so the idea here is that we'll keep antibodies around long-term that can actually be used to quantify roughly how much immunity a person has against a particular antigen that again would be attached to a particular microorganism. So we can do titers to figure out how much protection we have against certain diseases. I want to point out that there's going to be the initial response that will be very different because with the initial response or primary response, again, we're going to have this latent period. With the latent period, we have not quite yet matched up and started producing now antibodies against the antigen. And so the antigen is going to again be allowed to be around for a longer period of time because we're not pumping out antibodies to react to it yet. So this can again allow for this delayed response the first time we see a particular antigen. But once we've seen the antigen at least once, we should now be prepared to fight that thing again more effectively with again greater antibody synthesis and response. This would again allow for a peaking of the titer that allows for again long-term protection. So as we've always said, exposure helps lead to greater immunity. And that's exactly what this image is illustrating. Notice again the first time we're exposed, there's this latent period where we're not really producing antibodies because we haven't really matched everything up yet and started making the antibodies. So by the time we start making antibodies, notice we do a pretty decent response, but notice there's differences in concentration between IgM and IgG. You recall IgM was again used for agglutination and great at grabbing up all the pathogens that were allowed to reproduce during this latency. But again, IgG then will come in and finish up cleaning up the fight. The idea is IgG is great for long-term protection. But notice even what happens after that first encounter or primary response. IgG levels even go down to basically nothing. But there will be more memory cells that will stick around for the future encounter. So after a period of time, whenever we're exposed to the antigen again, notice how quickly we start producing antibodies. And notice now we shift a lot away from IgM and instead focus on IgG. And it's important to note there, notice that IgG levels never really go back down, at least not for a very long time. The idea is now that I've seen this a second time, there's a chance that I could see it a third time or a fourth time. So I need to keep more antibodies on hand to eliminate the threat before it gets too serious. So each time we again have these repeated exposures, we expect immunity to be pushed higher and higher and higher. And that's where things like boosters come in and vaccination. So let's move on now and talk about the 4 types of acquired immunity. They really fall into 2 categories, natural immunity and artificial immunity. With natural immunity, you're going to be exposed, or someone is, to the actual pathogen. So again, that exposure can lead to the development of, again, antibodies. Whenever we talk about artificial immunity, this is the purposeful introduction to elicit a medical procedure. And so again, artificial immunity, we're not seeing the exact pathogen being introduced to the patient. So let's go through them now in greater detail because each one of them has an active and passive version. Active natural is straight up picking up the pathogen and getting sick. And as a result, again, your body then develops immunity for that pathogen for future encounters. Passive, on the other hand, like the image is illustrating, is something like breastfeeding, where your mother again actively fought against the pathogen, but is giving you now her antibodies to protect you until you can develop your own active immunity. The idea here again is passive is short term, active is your own and typically long term. So let's look at the artificial example. Artificial active is vaccination, where we introduce the pathogen into you, but you're making your own antibodies and your own defenses against said pathogen. While passive, on the other hand, is we're giving you someone else's antibodies, again from their own previous encounters, that is offering you some short-term protection. So again, active is long-term and your own protection, passive is short-term, but again, sometimes necessary to buy time until you can develop your own protection. So let's talk some about the artificial active immunity that's really vaccination. Vaccination is the introduction to potentially a pathogen, but it's not really pathogenic. The idea is we're tricking the body to think that it is being attacked even though it's not. But the results are still the same, and the production of antibodies and immune response that will be powerful enough to fight this infection if we actually do encounter it. Again, since this really isn't the virulent thing, you're not actually going to get sick. But whenever it comes time that you do see the virulent thing, you're ready to fight it. And that's why this is an important technique. So vaccines are very necessary, but there are different types of vaccines out there. We have whole cell or virus vaccines, which gives you the whole thing, or parts of organism preparations, which again will just be pieces or antigens from a particular bacteria or virus. The idea here is the more like the real thing it is, the more effective we prepare the body to fight it in the future. And so the idea is we like whole cells or viral vaccines because again it offers us greater protection. But sometimes these things are potentially dangerous, and as a result we will only give you parts or pieces, or only certain features of the cell are actually dangerous, like toxins. So naturally there are lots of things to consider. How do we want to fight? What antigens do we want to protect against? How effective do we want this basic vaccine to be? How easy should it be administered? How safe is it? And how cost effective is it? So ultimately, we should have the lowest amount of side effects and toxicity so that we're not hurting people. But we want to expose you to as much of the natural thing as possible. And so the idea here is we got to find a happy medium that stimulates both B and T cells and offers long-term protection. Ultimately, we do not typically like lots of doses or boosters, but ultimately that might have to happen. And overall, we want things to be as cheap as possible and last as long as possible. Whenever we talk about the different types of vaccines, we have again the whole cell or virus vaccines. Again, these things can be killed vaccines, that are the whole thing, but are not going to be actively capable of causing infection. But we could also have the live attenuated vaccine, where it is the living microbe that can reproduce, but doesn't have the features that allow for severe disease. And as a result, again, the more like the real thing it is, the more we see better results. So live attenuated vaccines are actually very effective. But killed vaccines are much safer. Again, we can use subunit vaccines where we'll just give you parts and pieces from a particular bacteria, or again, products that we're concerned with like toxins. And so the idea by giving you just those things that we're concerned with is you'll develop immunity against those things, those dangerous features instead. But overall, again, these tend to be less effective and will often require more boosters over time. So one of the things that we've been capable of doing is actually combining several vaccines into one dose. We call these the multiple disease vaccines. So things like diphtheria and tetanus, that's called the DT vaccine. Or the DTaP or Tdap, that's again the diphtheria, pertussis, and tetanus vaccine. We also again have different forms that again may be multiple forms of hepatitis, like the Hep A and B, that's the Twinrix. The point is we can actually combine lots of different vaccines to create single doses that again people can get without being stabbed multiple times. Another important one I want to draw to your attention is the measles, mumps, and rubella, also known as the MMR vaccine. Again, a very important vaccine that we need, but may have come under some serious fire whenever the anti-vaxxer movement started up. So let's point out that we're always in need of new vaccines. There will always be new bacterial, viral, protozoan, and fungal diseases that we can't treat effectively with vaccines. We're trying to get there, and we can eventually get there if given enough time. The idea is, again, we have to tackle these, and there's lots of variables in play, so time is what's necessary to allow this to actually happen. Route of administration is another thing that we should point out. Route of administration allows for ease of administration. And so the idea is many vaccines are going to be shots that can be subcutaneous, intramuscular, intradermal. But again, most people don't like getting shots. And as a result, we like oral and nasal vaccines, but there are not many of them available. So whenever you get vaccines, be prepared to get stuck, because honestly, those are how most vaccines will be presented. An important thing we should talk about here is an adjuvant. An adjuvant is going to be a special binding substance that's added to some vaccines. This is ultimately going to enhance the amount of immune response, which allows for greater immunity in the long term. So it's not uncommon to see things added to vaccines to encourage the effectiveness of the vaccine. A good common example here is allium, which is aluminum hydroxide salts. Again, by adding these, we get greater immune response, which means we have to give fewer shots and still achieve great levels of protection. But there can be, again, side effects that come from vaccines. One of the most common things that we will likely see is things like inflammation or even fever. But again, inflammation and fever are things that are good to see because it's illustrating the body thinks that it's being attacked and as a response is trying to fight the invasion. Bad things that we don't like to see include things like allergies. Allergies can happen, and just like we discussed with drugs, any chemical could potentially elicit an allergic response. So it's important to hang out a little bit after you get a vaccine to make sure you're not having an allergic response. There are some super rare things that can happen, where again we can have a situation where we cause harm by vaccinating. Things like panencephalitis and measles vaccines have happened, but again they're very rare. Another example is back mutations and polio vaccines, where again we give you the live attenuated virus. that then back mutates and becomes the real poliovirus, resulting then in a virulent infection that could hurt a person. But as I said before, this is very, very, very rare. And the idea here is we routinely don't see this. And as a result, again, vaccines will save far more lives than they will ever take. Vaccines have actually come under fire a lot in more recent years. The idea is vaccine safety has been brought into question. I did just talk a little bit ago about this idea that vaccines are not completely safe and that there are risks, but they're very rare. But there have been connections made to vaccines that don't really stand the test of scientific scrutiny. And as a result, there are people out there that are getting bad information and making very bad choices that prevent them from being protected against diseases that we currently can fight. So one of the biggest concerns here was autism and the MMR vaccine. Ultimately, again, early on in the 2000s, we saw this publication come out that illustrated that there may be some connection between autism and MMR. But again, that study was refuted again and again and again. And even again in 2011, the Institute of Medicine published the results of a comprehensive exam of childhood vaccinations and showed no connection to autism and MMR. But yet people still point out that MMR and autism are tied together, and they're not. At this point, again, science has repeatedly shown that vaccines can and will be very effective, but also overall very safe. Again, vaccines save lives, and it's important that people become vaccinated to protect everybody around them. Because again, the person most likely to die from these style infections are those that cannot protect themselves. Those individuals that are so immunocompromised that they can't get vaccines themselves. And again, it's important that everybody does their job to protect those that cannot protect themselves. Since there was these unfounded fears over vaccine, people stopped vaccinating for lots of routine diseases, things like measles, mumps, diphtheria, polio, typhoid, even whooping cough. And as a result, we've seen huge amounts of infections that were once controlled showing up again in places like college dormitories, again, religious communities in which anti-vaccination is common, even in places like large-scale gatherings like the Super Bowl. The idea here is vaccinations are necessary to protect the whole population. And again, vaccinations can help save you from things that could potentially kill you. So get vaccines. Herd immunity is a very important thing that we've already introduced. The idea that if individuals protect themselves through vaccination, there's no place for the bad thing to live. And as a result, whenever some people can't get vaccinated because they have underlying medical issues, they're also protected because there's no place for the bad things to reside. But if people become basically breeding grounds for these pathogens because they fail to get vaccines, those pathogens can be then passed on to the people that could not become vaccinated, and those people are likely to die. And as a result, again, a person's actions and failing to get vaccinated even though they could have resulted then in the deaths of individuals that could never become vaccinated because of underlying health issues. Again, so your choice is really not your choice. Your choice affects many more members of our herd than you realize. And you should be conscious that if you do decline vaccination, you are putting not only yourself at risk, but also those that are relying on you to protect them. So hopefully this is raising a lot of questions of who and when should get vaccinated, right? And good news is the next few slides are going to help you understand that there are lots of things that you need even well into adulthood. Vaccines are not just for children. As you can see here, here's the list of the suggested basically schedule of vaccination, basically from birth to 6 years. This is again something that should be followed to ensure that the child has a safe ability to fight off pathogens they may encounter as children. Here we can see the recommended immunizations for children 7 to 18. The idea is there's still lots of them and there's a schedule to it. Again, active visits and active vaccinations are very necessary in protecting your children. But something else you should understand is vaccines don't stop at the age of 18. Vaccines should continue through the rest of your life. And so here's an illustration from the CDC that suggests again when you should get each of the different types of vaccines that are available.